Welcome to the deep dive. Today, we're looking at something really interesting, maybe even game-changing in Alzheimer's research. It involves nanotechnology.
That's right. We're drawing from a new study just out in the Journal of the American Chemical Society.
Okay, so Alzheimer's. We know it's this devastating neurodegenerative disease, cognitive decline, memory loss.
And for a long time, the focus has been on amyloid-beta peptides, these proteins that build up in the brain and form, well, plaques.
Exactly, those plaques. But this study, it takes a different angle, right? The mission here seems to be getting ahead of the problem.
Precisely. Instead of breaking down existing plaques, the idea is to use tiny nanostructures to target the amyloid beta before it can form plaques.
Nanostructures designed to trap amyloid beta like microscopic nets.
That's a good way to put it. They're specifically engineered for that purpose.
So the study talks about, let me get this right, trehalose conjugated peptide amphiphiles, TPAs. That's quite a mouthful.
It is. TPAs, yes. Basically, these are molecules designed to self-assemble. They come together to form these nanoscale fibers. Really tiny, thousands of times smaller than a human hair's width.
And these fibers are the nets.
Kind of like microscopic Velcro, actually. They're specifically designed to latch onto the soluble forms of amyloid beta.
Soluble forms. You mean the individual pieces before they clump?
Exactly.
Yep.
The monomers and oligomers.
Yep.
We now think these smaller floating forms are actually highly toxic to neurons, maybe even more so than the big plaques.
Okay, what about the trehalose part? What's its role in these TPAs?
Right, trehalose. It's a type of sugar, and it's known to be pretty good at stabilizing proteins. It also seems to help neurons survive.
So they added it to the mix?
They coated the nanofibers with it. The idea is it makes the fibers better at grabbing onto the amyloid beta, boosts their trapping ability, gives them extra stickiness, if you like.
Makes sense. So the big question, did it work in the lab?
Well, the initial results are definitely promising. The TPAs significantly cut down how much amyloid beta got taken up by neurons in their tests.
And the effect on the cells?
Cell survival improved by over 50% compared to the controls that didn't get the TPAs.
Wow, 50% is, well, that's a substantial improvement. But I assume it's not that simple. There must be challenges.
Oh, absolutely. This is still very early stage.
Yeah.
There are some major questions, like can these nanostructures even get into the brain? They need to cross the blood-brain barrier, which is notoriously difficult.
Right, the brain's security system.
Exactly. And we also need to know if they might interfere with other important biological processes, unintended side effects.
And just making enough of them.
That too. Scalability, mass production, it's always a hurdle with new nanotechnologies.
The researchers themselves are urging caution, I gather.
Definitely. They're very clear. This is a proof of concept. It shows the idea can work in a controlled lab setting. It's absolutely not a cure at this point.
So lots more work needed. Rigorous animal trials, presumably.
Essential. That's the next big step, and it takes time and careful validation.
Still, it feels like a significant shift in thinking about Alzheimer's treatment, moving away from just cleaning up the mess of plaques.
It really is. We're talking about prevention rather than cure, or at least much earlier intervention.
Trying to stop the cascade before it really gets going.
How does this compare to, say, the antibody drugs we hear about, like aducanumab or lecanemab?
Those drugs primarily target the larger amyloid aggregates, the plaques themselves. This TPA approach goes after the smaller soluble species before they aggregate.
So potentially more effective, maybe, in the very early stages.
That's the hope. By targeting these possibly more toxic, smaller forms, you might prevent the damage earlier. It's like stopping sparks before they start a fire instead of just putting out the flames.
Could these approaches eventually work together? Maybe. TPAs early on, antibodies later?
That's certainly a possibility down the line. Nanotechnology holds a lot of potential in neurodegenerative diseases, but again, as the experts always stress, rigorous validation is key. We can't jump ahead. So potential for TPAs to complement existing therapies, or maybe even one day a preventative measure for people at high risk.
It's conceivable, yes, but that's looking quite far into the future.
Okay, so wrapping up our deep dive into this JCS study, we've got a novel nanotechnology strategy, using these TPA nanofibers as tiny traps for soluble amyloid beta.
The goal being to intercept Alzheimer's much earlier, before those damaging plaques fully form. Promising lab results showing reduced A uptake and better neuron survival.
But, and it's a big but, this is proof of concept, early days. Significant hurdles remain, like crossing the blood-brain barrier and ensuring safety.
Absolutely. A long road ahead for research and development.
It does make you think, though, considering the potential of nanotechnology to tackle these really complex biological challenges, How do you balance that potential against the very long timelines, the setbacks, the sheer effort involved in developing actual medical breakthroughs? Something to ponder.